Thank you for coming to see my poster. My subject today is phenotypic plasticity for temperature in reptiles. In brief, I have used Madagascar ground geckos to observe gene transcription and chromatin accessibility under different temperature conditions, showing that short-term and long-term temperature exposure leads to different molecular responses. First, I'd like to talk about some background on this study. Ectosaps respond to environmental changes such as global warming in a variety of ways. Among them, phenotypic plasticity, in which an individual's response changes in response to the environment, is thought to be counteract genetic adaptation by reducing selection pressure. So, understanding the mechanisms of phenotypic plasticity can help predict the potential for evolutionary adaptation to environmental change. An important factor in the response to temperature is the duration of exposure. For example, in mammals, short-term heat stress induces a heat shock response at cellular level and changes in heart rate, whereas a long-term temperature response reprograms gene expression and alters metabolic pathways and homeostasis. However, the effect of different durations of temperature responses have not been clearly distinguished in ectoderms. And therefore, the purpose of this study was to determine the differences in molecular responses to different temperature exposures over short and long periods of time. To this end, gene transcription and chromatin state were observed under different temperature conditions. Next, I'll describe the target species and methods. The target species is the Madagascar ground gecko, which has recently attracted attention as a new model species for reptiles. This species is a nocturnal gecko that can be easily kept and bred, and its genome has been sequenced and genome editing is possible. Therefore, this species was used in this study. The individuals were then separated into a total of 5 temperature conditions, 3 conditions of 4 hours as short-term treatment, and 2 conditions of over 50 days as long-term treatment. After which, the livers were sampled for ATAC-seq and RNA-seq. The analysis procedure is shown here. And this allowed us to observe changes in gene transcription and chromatin state in the short and long-term temperature change. In the results and discussion session, I will first discuss the observations of gene expression by RNA-seq. The results of the principal component analysis of total gene expression showed that the short-term temperature-treated samples at 30 and 37 degrees and long-term temperature-treated samples at 30 degrees had different expression profiles. As for the differentially expressed genes, over the half of differentially expressed genes due to long-term temperature treatment did not overlap with differentially expressed genes in the short-term temperature treatment. And in addition, the functions of differentially expressed genes were different between the short and long-term treatments. Many genes involved in non-heat stress responses, such as translation and protein refolding, were altered in the short-term temperature treatment, whereas genes involved in immunity and nutrient metabolism were altered in the long-term temperature treatment. Then I will talk about the results of chromatin state detection by ATAC-seq. And this figure shows the changes in expected occupancy of transcription factor binding sites with temperature treatment from 25 degrees control, with darker blue indicating a decrease in expected occupancy with temperature treatment, and darker red indicating an increase. As can be seen from this figure, many transcription factors showed changes in occupancy in the opposite direction between short and long term. Transcription factors whose predicted occupancy changed significantly in both the short and long term are shown here. Of these, CEBP family transcription factors are involved in cell differentiation, metabolism, and immunity, and have been reported as temperature-responsive transcription factors in several other studies. It is possible that changes in these and other families of transcription factors produce different short and long-term molecular responses. In conclusion, the results of this study indicate that different molecular responses occur with temperature treatment for different periods of time. A short-term temperature stimulation of a few hours altered non-heat shock responses and circadian rhythm-related genes. Whereas long-term temperature treatment, temperature acclimation of tens of days, altered immune function and nutrient metabolism. And this switch from short-term stress response to long-term acclimation may be achieved by larger-scale changes in transcription factor occupancy. Thank you for your attention.